Okay, welcome back. So our third speaker is Dr. Bikash Ghimire. So this will be this hour or less than an hour. We'll be focusing on ornamentals, starting with pathology, then entomology. Dr. Bikash Ghimire is basically replaced Dr. Jean Woodward. So that is basically the update from for the industry. It's a big update, I guess. Okay, a little bit about Dr. Ghimire. So Dr. Ghimire is an assistant professor and extension specialist in ornamental plant pathology at University of Georgia. Dr. Ghimire specializes in disease management of ornamental plants, focusing on integrated strategies that combines chemical, biological, and host resistance approaches. His research includes fungicide resistance, biofungicide evaluations, and the use of advanced genomic tools to study plant pathogen interactions. Dr. Ghimire works closely with growers and industry partners to develop sustainable solutions for ornamental plant health. Dr. Ghimire, the floor is yours, okay?
Awesome. Thank you, Dr. Joseph. Let me share the screen real quick. Yeah, I think I need to share my screen too. I'm going to share it now, and then can you see my slide in the
Yes, we can.
slideshow mode?
You're good to go.
Good. Awesome.
Yes.
All right. Thank you. Thank you for having me, and it's a pleasure and an honor to be here. And I would like to talk about the industry update on ornamental, diseases. But before that, I just want to give you a quick overview of my background, which started back in the fall, where I completed my bachelor's degree and then joined for a few years in a government research institution. And after that, I moved to Washington State University to do my master's in crop science. Then I moved in 2017 to University of Georgia, and I never left because I completed my PhD. And right after that, I was a postdoc for like 3 years. That was back in Griffin campus. And eventually, in August of 2025, this year, I joined as an assistant professor here at UGA, basically replacing Dr. Jean Woodward. So with that, just the outline of my talk will be, I'll talk a little bit about the plant pathology basics, ornamental horticulture industry in Georgia, some of the common diseases that we have in ornamental disease, plant, and my extension and applied research program at UGA. So as a plant pathologist, we love to talk about the disease triangle all the time. So, but before that, I would like to just draw your attention for on the left of the slide where I have this categorization of major plant pathogens, and 80% of the disease that we have in the field are due to fungus or the oomycetes, and 10% are due to bacteria, and we have very low proportion of virus and nematodes, which are 5% each, but they are still a big problem sometimes. So now coming back to the disease triangle, for a disease to happen, we have three important components. Host that need to be, the susceptible, pathogen that need to be virulent, and the environment that need to be conducive. So if we have all three, the factors that are perfectly there, then that's when we have a disease, basically plant disease in this case. Again, as a plant pathologist, I want to give a couple of examples, which is of historical significance on why we even care of this plant disease. Because I'm sure like a lot of you have heard about the chestnut blight, which is a fungus disease caused by Cryphonectria parasitica. That was a big problem in 1900s because we used to have more than 50% of our, the eastern hardwood forest covered by chestnut. But all of a sudden, we lost almost 100% of this chestnut by 1950. And if you see the map on your right, you can see how the disease was progressing from 1914 from the north towards the south, and every year it used to go down 50 miles down the south. So that's how the disease was so much of the problem. And I'm bringing this point because I'm going to talk about some of the new disease that we are facing in Georgia, and then just want to have that background that why we need to understand new pathogen and diseases. And here's another example I want to give again about the sudden oak death, which most of you might have heard. This is caused by oomycetes, which is fungus-like, but actually not a fungus. It's a Phytophthora ramorum, and it was first identified in California in 1995. And as you can see in the map, so the pathogen was diagnosed as ramorum in 2000, and that killed almost 30 to 45 million trees in California and Oregon. So that was a huge loss. And as you can see, that cool video on your right, that how this pathogen had this cup-shaped sporangia that bears, that's a fruiting structure of the pathogen, and that's where it was dispersing all these zoospores, which is water molds that needs water to swim and cause the infection. So that's how we understand the pathogen and the disease. So now with that example, let's talk about the ornamental horticulture industry in Georgia. As all of you know, this is the fourth largest commodity group in Georgia. We have the farm gate value of over $1 billion, and then nearly 67,000 people are engaged. So this is a big industry. And on your right, you can see the graph, and this is the best part that I like, because as you can see, the y-axis, you have this, the year from 2001 to 2022. And as you can see, starting from 2013, 2014, we started seeing that uprising the ornamental horticulture industry. And really it's booming, and then there is tremendous opportunities. But at the same time, we have new diseases also that is we have to deal with. So when I started this position, even before that, I was talking with several, the stakeholders, county agents and industry folks, just about like what are the major problems that we have in state. And then what I come to find out was I narrowed it down into three different categories. So first one is the root and crown diseases. And then this could be caused by oomycetes and fungus. So when you say oomycetes, that could be Pythium or Phytophthora. And for the fungi, it is mostly Rhizoctonia and Thielaviopsis. But we have a whole lot of, other pathogens like Fusarium or Verticillium that could be a problem. Verticillium is really not a problem in Georgia because of the soil pH, but it could be a problem in other parts of the region. So as you can see, root and crown rot are the major problems in the greenhouse and nurseries. And I'll talk a little bit more about these diseases later in my slides, but that this disease can be a problem in the landscape and woody ornamental plants also. So the next category I have is the foliar disease, where we have leaf spot. And leaf spot could be caused by several different fungi. I have listed Sarcospora, Alternaria, Entomosporium, and Coletotrichum as some of the major culprits. And just to remind, or just to let you know that we have been doing some kind of research on Sarcospora leaf spot in crepe myrtle. Actually, Dr. Gene Woodward was collaborating with a breeder here, John Reuter, and then trying to screen some of the cultivars in the Blairsville Mountain Research Station a couple of years ago. So fungal leaf spot are both found in greenhouse ornamentals and landscape plants. The next category I have here is blight and plant virus diseases. So on your far left, you can see Passolar needle blight in Leland cypress. That's a big problem here in Georgia and in the south. And then in the middle panel, you can see Boxwood blight, which is caused by Calonectria. And I'll talk more about this disease later on because this is something I'm interested to work on. And then on your right, you can see plant viruses, for example, rosette virus. So blight and plant virus are devastating our landscape plants. And then we have some other diseases like caused by bacteria and nematodes. For example, we have Xanthomonas, Pseudomonas acidovorax that can cause leaf spot, or we have a problem with the root knot nematode. But it's not that prominent as compared to the fungal diseases or the plant viruses, but it could be a problem. And then just to be vigilant on that. So more about the virus diseases. So this is actually increasing, and then this is again rosette virus. So as you can see, this is a big problem in rose, and this is transmitted by iridophyte mites. But there are other pathogens like insects like thrips and aphids that can transmit several other different viruses. For example, this lily yellow mosaic virus and yellow streak virus. As you can see on the right, it is transmitted by aphids, and then a lot of time these can also be transmitted by grafting or mechanical transmission or propagations. Actually, there is no cure for these plant diseases, virus diseases, because you cannot basically spray any, the fungicide, but what you can do is control the insects, because that's a vector. That's how it gets transmitted from one plant. So basically, healthy to unhealthy plants to healthy plants. So that's where we need to be aware, controlling the monitoring for the insect population, and then using right insecticides. And then if you have infected plants, all you need to do is discard them so that you can reduce the spread of the pathogen. So with that, a little bit of background, I want to now talk a little bit about my extension and applied research program here at UGA. As I started in August, it has been like my third month. So really looking to build up my program. So I need to mention that I'm an extension specialist. I have 85% extension role. So I basically love to work outside with the county extension agents, try to understand what's the problem out there, and then basically provide them science-based findings through my extension program. I love to engage and educate, as I said, like stakeholders, industry clientele. I do a lot of like site visits that I'm planning to do in the future, and just to understand the problem so that I can take that back to my research program and formulate my program accordingly so that it can help solve the problem in the field. And then also I look forward to disseminate some kind of the scientific information through traditional and digital platform, extensions of bulletin through UGA, and also attending several conferences and webinars like GTBOB that we have now, or Southeast Green, Urban Ag Council, the meetings and all that thing. And then one of the other important aspects, and let me tell you, this is where I was on your bottom left. You can see I was with Dr. Jean Woodward, who was in this position before, as I mentioned. So I got to meet her in the Southeast Green Conference, and then I was actually getting a lot of valuable insights from her, because she has been here in this role for nearly 20 years. And then the other important extension role I have is managing the recommendation of the major diseases that are the plant samples that come to our disease clinics. So I don't know how many of you know that we have a plant disease clinic in Athens. That's where we receive samples, disease samples. And then we have a plant diagnostician who will diagnose the pathogen or the disease, and then I'll be sending out the recommendations for ornamental and commercial landscapes. So just to give you an overview of the plant disease clinic, on your left, you can see there's a submission form that you can fill it out, and you can take help from the county extension agents whenever you encounter with problems. For example, if you're a landscaper, then if you have some of the homeowners struggling with some of their plants, then you can have that plant sent with this form to our disease clinic, and then eventually the clinic will process the sample free of cost if you are a commercial, the clientele. So there's no charge, and then eventually we'll diagnose the pathogen and then provide the management recommendation that best suit your conditions. So the next important aspects I want to undertake is doing the survey and surveillance of existing diseases. So when I say existing disease, as I already mentioned, that we have a big problem of root and crown diseases, and then it could be due to fungi that cause a dry rot like Rhizoctonia and Thielaviopsis, or it could be oomycetes like PTM and Phytophthora. And it's a big problem. That's why I'm repeating this again. As per our experience from the plant disease clinic here in Athens this year, majority of this like a root and crown rot disease was either by Thielaviopsis or by Phytophthora. So that's where we have been observing a lot this year. And then there are chemical management options. And we need to really understand if it is fungus versus oomycetes, because some of the fungicide that you apply for the oomycetes, for example, mefenexam and metalaxyl, doesn't work or have not that efficacy as for fungus. And then so does with the fungus pathogen that you have to spray, or you can spray diamide fungicide, thiophenol methyl, or azoxystrobin, and not every of them will work for oomycetes. So you need to really diagnose the pathogen so that you can tailor your management recommendations. And a lot of time I would say these are due to the root and crown rot disease are due to bad, or I would say maybe not appropriate cultural practices, that because I have been seeing a lot of, especially when I was doing a nursery visit, and then I saw they were either over irrigating, or they had, very compact and no air circulation and all kind of things that was actually cultural practices that could be modified, and that will help to reduce the pathogen load actually. So if you have a crown rot, the problem, then we have a very, quick techniques of identifying the pathogen. So we have a immunohistory test. So that is developed by AgDia. And then all you have to take is you have to take an infected root or a crown, and then you put it in that Ziploc bag that has this buffer, and then it will give results in as quick as 5 minutes. And then if you have two bands that you can see in the figure, then it will confirm that you'll have Phytophthora in this case, because this immunohistochemistry is specific to Phytophthora. So that's how we test for these different diseases of the pathogens. So now let me talk about some of the emerging disease that we have in the state. And I'm going to talk about two of them that I'm interested to work on. Basically, vascular streak dieback. I don't know how many of you have heard of this. This is a problem that is going around in the southeast and in Oregon, caused by a fungus, Ceratobacillium species. It's mostly in the woody ornamentals. It's a major problem. I would say in the next slide, I think it's in the red maple, dogwood, and the red bud. But it was first reported in Tennessee and North Carolina in 2019, and it has been at least reported in 11 states. And as you can see in the left, that in one of the nursery in Virginia in 2022, they had 90 to 100% of the dogwood rootstock or the nursery plant that they had to throw away. So it's getting a big problem. It has, and you can see in the map, Georgia is untouched, and all other surrounding states are here. So based on my conversation with Dr. Woodward and a few of the nursery visits and landscape companies, so I suspect that the pathogen is already here, and that's where we need to work on. But as I said, it can cause disease in more than 34 different woody ornamental plants, and it does wilting, leaf scrotching, and yellowing, and vascular discoloration. And you can see the branch dieback as well. So there's research undergoing across different universities like Tennessee State University, Virginia Tech, and all that to understand the pathogen and confirm the pathogenicity test. And that's what I'm teaming up with some of the faculty members from Tennessee State University and Tennessee Department to work on this disease for next season. So all for the management option, we have cultural and sanitation practices. There are chemicals and biological, the fungicide that can be applied, and there are some resistant variety actually that is just based on the research findings that they are still testing for some of the varieties if it is really resistant to the disease or not. So the next one I have is lethal bronzing disease. So in palm, and then I got an email even before I started this position in August 1st. So I got an email there is a problem with the sabal palm mortality in Thomasville, which is near the Tallahassee in the state line. And that's where the clientele was sending some of these pictures, and then we didn't have the ability to test in our lab here in Plant Disease Clinic in Athens. So we routed them to send the samples to Florida, and then guess what? It turned out to be lethal bronzing disease. So it actually starts with the oldest leaves, and it progresses upward, and then eventually the younger foliage will also get infected, and it has a bronze-like browning of the leaf. That's where the name's called, the bronzing or the lethal bronzing comes. And then it was first reported in Savannah when it comes to Georgia back in 2024, last year, and it is caused by phytoplasm, which is a bacteria without the cell wall. And the interesting thing about this disease is it is vectored by plant hopper, and the pathogen is phloem limited. It lives in phloem, and then based on the findings that came out in this 2024 literature, the plant material was actually moved from west coast of Florida to Savannah, and in Florida they have been experiencing this disease since 2008. So this is not a new disease for them, but for us it's kind of new. And then it's really important to understand how the disease and the vector are moving from maybe Florida and then causing a new disease here in Georgia. And we have already reported a positive results from Savannah and from this Thomasville. So at least we have reported in two different areas. So for the management, I would say we have to immediately remove the infected plants if it is tested positive. And if the neighboring plant palms are tested negative, then you can actually provide antibiotic oxytetracycline SCL preventatively to manage the disease, and it has to be applied quarterly or 3 to 4 months. And the next one I have now is Boxwood blight, which is a big problem all the time. I'm sure you might have dealt with this pretty often. And then, interestingly, we didn't see much report in the disease clinic this year in Athens. And part of that was the clinic was closed because of we didn't have any diagnostician. So the new diagnostician here in Athens started in August 1. So that's maybe the reason that we didn't see much of the disease sample this year. But all you need to do to identify this disease is look for the defoliation. So it will massively defoliate the plant, and then you can see the black stem lesion. That's what the characteristic symptoms for Boxwood blight. And then I really want to work further understanding the pathogen diversity and understand what is the species, what are different diversity that are prevalent in this specific pathogen. So that's what I wanted to understand here in Georgia. And then we are collaborating with Dr. Rooter and Dr. Yadav here in UGA to screen some of the commercial cultivars and breeding lines so that we can come up with the disease-resistant cultivar in the long run. So that's the long-term goal that we have. And for the management, so we have to remove the infected plants and the lower branches. Really, you need to disinfect the pruning tools and gloves, and mulching can be helpful sometimes just because it can help to act as a barrier for splashing the spores to disseminate. And then you just need to avoid like getting too wet to the foliage or avoid the overhead irrigation if it is not necessary. And then we really recommend to plant resistant cultivars. Mostly Korean types or Korean boxwood are more resistant compared to American or the dwarf English. And there are some chemical fungicides that can be applied every 7 to 14 days: chlorothalonil, fludiaxonil, tebuconazole, and propiconazole. But all we need to do is stop the spread of the pathogen, and that's where pests could be sometimes problematic. So we want to make sure that the pests are out of the garden. And then we have two other diseases in boxwood that we commonly identified in plant disease clinic this year, which is boxwood dieback and Volutella blight. So for the boxwood dieback, it is called Bicolate trichum. It's kind of new disease here in the state, reported in 2015 in Louisiana. So it causes the dieback symptoms, and it is often confusing with the boxwood blight, Volutella blight, or the Phytophthora rooter. So it's always like it looks similar, so it's very much confusing. And then I get calls from some of the county agents or even from the clientele that how can I identify the pathogen? And then they send me pictures, but it is very difficult. And I'll ask them to send the plant sample to the clinic so that we can accurately diagnose. And we have this Volutella blight where the diagnostic feature is we see the salmon color fungal mass underneath of the leaves, and leaves are still intact in the plant, versus in the boxwood blight, leaves are all in the already defoliated. So I believe this is my second last slide here. So this is the another areas that I want to really work, which is cut flower. So this is a good industry that is emerging in the state, and there are tremendous opportunities out there, and there is a strong demand from the stakeholders. So I am collaborating with a few of the UGA researchers, trying to put a proposal, and then we really want to survey for the major disease that we have prevalent, either in the standing field and the post-harvest condition in the cut flower. And then the major problem, guess what, is botrytis blight. That's what we have been constantly fighting for. So my future projects will look like some of the fungicide screening experiment, either with the chemicals or combination of chemicals and biological products, and then also doing some kind of the post-harvest treatment to better manage the different diseases out there. So overall, my program will always be the growers' demand, like the growers and the industry driven based on their needs and the problems they are facing, and then it will be based on the collaboration with different researchers and scientists at UGA and outside, so that we can maximize the impact to our green industry in Georgia. So with that, this is the last one where I want to just emphasize that we have a Georgia Pest Management Handbook, and then I always recommend all of you to refer to this book whenever you have to come up with the chemicals and biological fungicides, what are there out there for certain diseases or the host. And then we have a big collection here, like a good information out there. It is updated every year, so it's a good resource out there, so to refer. So I think with that, I would like to thank all of you, for listening to me, and then I'll be happy to take any questions. Here's my contact information, and on the right left, you can see the the QR code. You can participate in a sort of like a couple of minutes of survey, which really helped me, to understand my program, how I can, first of all, like the presentation that I gave you, how you like, and then also I have towards the end like a major disease that you have faced in your, the facilities or landscape companies or wherever you are. So I would love to hear about the problems that you are facing so that I can come up with that in my program for the years to come. So thank you.
Hey, thank you so much, Dr. Ghimire. So because there is one question, there is another question about insecticide issues insect issues in viburnum that I will address. This question is, if you have a hedge of boxwoods and several other shrubs have blight, is it best to go ahead and remove all of them? Should the soil be treated before replanting? That is a question.
Is that for the
Excellent
blight?
question.
Can you repeat the first part?
Yes. Yeah, the first part is if you have a hedge of boxwoods and several other shrubs that have blight, okay, is it the best to go ahead and remove all of them? That's the first part of the question. Second part is, should the soil be treated before replanting?
So I have found that it depends like how much of the is how first of all how older is your plant in the landscape, and then how big the problem is. Because I was getting a call, or actually I got an email, and then it was from the homeowner. She has like the entire haze of the boxwood, and then that was so much devastating. At some point, if it is too much to do the chemical management or pruning, especially in this case, if it is too much to prune, then it might be worth it to replace if it is really heavy and infected. So it all depends. I would say like prune for a year and see how much it will come back, because there are evidence that once you do a proper pruning, then it will come back. But don't expect to have a beautiful the hairs in a year or two. It will take some time. So it all depends on how much of the infection you have and what is how much you are attached with your plants and all that thing. So it's a kind of tough decision, I would say.
That's good. Thank you so much. For the sake of time, let's move on. So,
Absolutely.
Vikas, thank you so much for excellent talk and good luck for all your research program. And I know you just started, so I hope in the years to come, we're all looking forward to. Okay, thank you.
Absolutely. Absolutely. Thank you for having me, Dr. Joseph.
Sure. Okay, so let's move on with an entomology.
All right. Well, it's my pleasure to take a minute here just to reintroduce our host and our fourth speaker for today, Dr. Shimat Joseph. Dr. Joseph, he's our associate professor of entomology at the University of Georgia here on the Griffin campus. His research and extension programs focus on turfgrass and ornamental entomology, while he's addressing pest management issues in urban landscapes, nurseries, sod production, and golf courses. He emphasizes integrated pest and pollinator management, aiming to develop environmentally responsible strategies that protect beneficial species while controlling invasive and emerging pests. Dr. Joseph earned his PhD and his master's in entomology from the University of Georgia, and he has a wealth of prior experience as an IPM advisor while he was with the University of California. And Dr. Joseph, the floor is yours.
Hey, thank you so much, Ruchi. Looks like I need a permission to do that. Maybe now. Yes, now I can read. Wonderful.
All right. I'm seeing it.
Great. So before I start, I just want to address this comment. So this question is in the southern coastal areas, we are having a major problem with whiteflies on viburnum. Do you give insecticide advice? We have used Dominion and Bifint, but insects migrate from other properties after. Yeah, it is a pretty dire problem, because once the cotton is cut or harvested, all the whiteflies from the cotton moves to everywhere. That includes into our yard or ornamental nurseries. So that is a problem. So if you're going into managing it, I would say Dominion is good. That's imidacloprid. That can be applied as a drench. It works better as a drench. It all depends on when you are starting to do the management. If you're going to do like for 2 months, you're looking at that down the road, then I think a drench will be a good strategy. It will be good as a foliar too, but it will be best as a drench. Bifenthrin is a contact insecticide. Yes, it will only provide a control up to 14 days. So if you want to manage it for 2 months or down the road, because it starts from sometime in I would say September, when the cotton winds down, you start moves to the ornamentals. Imidacloprid, that is a neonicotinoid. Those products, acetamiprid, that is Tristar, Aria, clothanidin, flagship, that is thiamethoxam, and all those products, neonicotinoids should work. Group of insecticide is pyrethroids, that is you already mentioned here, bifenthrin. Others are permethrin, deltamethrin, cyphalothrin, all those thrins towards end, the thrins, those should work also. Third group that you can look at is IGRs, insect growth regulators. That includes pedestal, nivaluron, if it is nurseries. Otherwise, is pyriproxyfen, that is distance, fulcrum, those kind of brand names, or brufacin, that is Talus. So these products will work. So if you do look at rotating it, and that should provide you some control for up to 1.5 to 2 months. So make sure you rotate all these chemistries, and you'll be able to control it pretty long. Okay. So that's my comment for that. And unfortunately, they fly and come back if the residues goes down. That's the nature of these whiteflies. Okay, so today I have about 15, 20 minutes. So I want to focus on an emerging pest issue in Georgia. So this is called two-spotted cotton leafhopper. Okay, that's what I want to talk. So this is my contact information up, in the beginning itself. I just want to give you. And this is my lab website. So if you are interested, let me move the slide. So Ruchi.
Hey, does it help if you click on the
Enter.
bottom left part?
Yeah, it's not moving actually. So let me do it one more
Yeah,
time.
that's what I would do too. Just break out of the share and share it again.
Yeah,
Sometimes if it's acting up on the PowerPoint, I'll just share the whole screen.
yeah. Let me see. This works. Here we go. Now I can do it. Before I go and talk about this pest, I just want to let you know that we have a website where we update about any emerging pests in our state. We call it as Turfgrass and Ornamental Pest Management. So if that happens, we write a blog article, and if you sign up here with your email and first and last name, you should receive an email, and that will let you know about the new information. This also includes information on diseases and also weeds like Ben and Dr. Alfredo Martinez. We're all right together. So it's not just entomology, but you will get emails. If you're interested in knowing about the pest issues, major pest issues, this is also a resource for you, where you can see here turfgrass pest and landscape and ornamental pest here, where you can click that and it lists all the different major pest problems we have in Georgia, so that you can learn the biology and how to quickly manage it. Okay. More. Two-spotted cotton leafhopper, that is an emerging pest. I would say this actually hit us this year. Okay. So we call it as two-spotted cotton leafhopper or two-spotted leafhopper or cotton jassets. It's basically leafhopper. Okay. And it is actually native to Africa, anywhere from Iran and all the way to Japan, in this whole range where these are naturally present. And they got introduced in 2013. We started seeing in Puerto Rico and the US Virgin Islands. And in last year, it was detected in Florida. And this year, we have in 5 states already. Okay. The problem here is cotton. It's a major problem in cotton. And we have seen cotton growers struggling to keep up with spraying whatever they can throw it. And that's what's going on. That also went on this year. And it's pretty serious. We don't know how they move around. There could be some possibilities like the storms that bring in from the East Coast could be from the Florida, East Coast, from the southern part of the Florida all the way up to north. That could be one maybe one way they're getting around. Another way is moving plant material that are already infested. So there could be 2 ways. We know that this is a pest on hibiscus because we are seeing them on different species of hibiscus, tropical hibiscus, that is, or Chinese hibiscus, that's Hibiscus rosasanensis, or the hybrid here, where Dr. Reuter, who's a breeder, Dr. John Reuter, he reported on this particular hybrid, and we are seeing the population on in Syriacus. that is Hibiscus syriacus. So they are a pest of hibiscus for sure. We know that for sure. I'll show you a few slides about that. Okay. I looked up, this is a new pest, so we want to know what's going on. So I looked up the literature, and this is what I found. So can be a problem on 27 known species of plants. That is already known. It's primarily Malvaceae family and Solanaceous family. It can be other plants too. So these are the major crops that we are seeing. You can see cotton, okra, eggplant, tropical hibiscus, Indian sorrel, sunflower, watermelons on cucurbitaceae, melons, cucumber, gourds, different kind of gourd, sesame, and also grapevine. This is what they say, what is reported in the literature as a reproductive host. What I mean by that is that when adults settle on this host, they can lay eggs and they can continuously develop on this host and become another adult. They can successfully complete a generation. That's what it means. And you can see some highlighted here: tropical hibiscus, sorrel, and pepino. Apparently, these are ornamental crops in Asia, and that's basically highlighted here. Okay, so I just want to show you that. And there are other papers that talk about hibiscus as an incidental host. In this case, they have seen them on the plant, doesn't necessarily reproduce or go through a generation. That includes poinsettias, croton, roses, gerberas, marigold, pepper, cilantro. There are other vegetable crops and that like tomato and pepper and mulberry and things like that. So the point is that within I said Malvaceae and Solanaceae are do important and cucurbits. Within that, there is so much difference there. For example, pepper and tomatoes, they are considered not very major problem because we have seen this year in the field where they are hammering and killing the eggplants and also the okra, but they are not even doing anything to tomatoes and pepper. So within the same family, they are differentially attacking and colonizing and causing issues. So there's something to know. So these are the host range on this pest. What it will do: yellowing, browning, and it will curl the leaves and cause necrosis, meaning the curled affected portion of the leaves becomes brown in color. That's what it says, and eventually the leaf will drop. I'll show you some pictures on that. This is on okra. As you can see, the leaves are basically stunted, right? The plant is basically stunted, and you can see it kind of curls upward in this case. And these are all the leafhoppers you can see over here in the picture. And with that feeding, the yellowing comes in, the leaves curls up, and eventually some areas become necrotic. That's what I say. People refer to as a hopper burn. That's the term used, meaning it will, if the population continues like that, the entire field will be like burned. Somebody put a fire on it. That's what it will look like. It will like burned appearance. And so that's what you say. That's a more extreme situation. But we have seen that extreme situation in cotton this year and in okra. So this is on cotton. You can see starts off with the yellowing here, and gradually the necrosis develops. And you will see in this case, the leaves is folding downward for cotton. And we see that. And also what another symptom we saw in cotton is that the new shoots are coming out, they're kind of clubbed together. They are not like expanding normally. So that is kind of a virus symptom, like a rosetted appearance. But there is no report of any virus involved or phytoplasma or bacteria involved. That's what we know to date. That more research needs to be done to figure out that detail, because often leafhoppers transmit viruses or bacteria. In this case, to date, we don't know about that. There is no reports of that, any transmission involved. So this is an example of the damage, how it looks like. We have seen on Chinese hibiscus or Hibiscus rosa-sinensis. As you can see, it can be a problem. So the problem we are concerned in ornamentals is we may not see necrosis or hopper burn, but even here in this case, you can see the feeding causes yellowing. That starts from the margin and eventually basically spreads all over. And I have seen places where the leaves basically drop off completely. So for us, this is an economic damage because our business is beauty and should look good, should be quality plants. And so this damage can be a quality issue and unmarketable situation for us, for our ornamental industry. This is another species of hibiscus, Hibiscus syriensis. If you can look here, you'll see this particular block is completely, the plants are yellowed. And you see that yellowing starts from the margin and all the way. And there's a lot of leaves fallen. I couldn't take that picture, but you'll see a lot of leaves fallen. They're all drop off after the yellowing. We didn't see any necrosis or burned appearance in this case, in this particular species of hibiscus. This is same variety. As I mentioned, the yellowing starts like that, and it goes all the way down, and the leaf drops. And here in this picture, you can see how it progresses and continues. I looked at two cultivars. So you can see this one cultivar here. This is barley. Another one is purple chiffon, or lavender chiffon, that's what's called. But you see there is a differential here. In this particular variety, the damage is really bad. And this one has some yellowing, but not as much as that. So maybe there is a varietal preference or varietal population dynamics there that we might need to work on in the future. So the reports from the Asia also talks about weeds. So there are many weeds these weeds maybe not very relevant for US, but you can see some of them are listed here. So just to keep in mind, especially the wild types of hibiscus and the fennel, Indian mallow, we have some here in Georgia. So now getting to talk about the insect itself. As you can see, the insect is very small. They are very tiny. They are about 3 millimeter in length. Okay, and the adults also has two spots here. That's very unique. We don't have any leafhopper with that characteristics. So if you see leafhopper about 2 to 3 millimeter long and there's two spots, that basically means that is an invasive. That's what we know at this point. Okay, there are potato leafhoppers about same size, but you will not see. But this one is not potato. It is the same species. The thing is, it takes, as they molt from 15th star to the adult, it takes some time. So, you have to give some time for the spot to develop. So as they emerge out from the 15th star, there won't be any spots. But with time, like you're talking about 10 to 15 minutes, the spots will appear. Okay. So I just want to say that because we observed that in the lab. So we know in Asia there is about 11 generations because they don't take that much time to grow from a complete one generation. These things are highly subjective to temperature and also relative humidity, just to keep in mind. So about 15 to 46 days, that's what the range that we are talking about to complete a generation from egg to adult in this case. So it starts like with eggs. Eggs are laid in the midrib or the veins that we won't see the eggs at all from outside because they punch inside the midrib area or the veins here in this region. And they lay about 20-25 eggs or 14 to 38 eggs, depending on what we have seen. So different papers talks about that. So there is a range that we see, but at least it says about 14 eggs for a maximum of 38 eggs that you can expect from a female. It takes about 3 to 4 days to hatch. Again, temperature is a big player here, and they go through 5 instars. So it starts with the first instar to third instar here. You can see they are small in size. They may not have any wing pads. So that's pad that we talk about. This is one example here, the wing pads I'm talking about, but only the size difference, small to a little bit bigger in size up to third instar. Then they molt into the fourth instar where you'll see a pair of pads here. Then moving into fifth instar, we'll see 2 pairs of pads here for that, and they're a little bigger in size, and they molt into an adult. Okay, and the whole time it takes is about 8 to 11 days in our summer. So with the high temperatures, this is what we'll see. So here is a picture of all the stage. We see them on the underside of the leaves. We never saw them in the upper side. So they colonize in the underside of the leaves, and you'll see all different stages at the same time in the summertime. So this is the first instars, second instars. You'll see the third instars there, fourth, fifth, and even adults. Everything together under the leaves. So this leaf is an okra leaf. Okay, where you took the picture. So I have a postdoc working in my lab, and she was, doing some surveys to understand this, what's going on. And her name is Dr. Sabrine Attia. So she did some preliminary studies, surveys to understand about this bug, this new pest in the nurseries. So here we could try to see some varietal difference. Two varieties that we sampled, we found more in one variety versus other, but we can't confidently say that across the board. So we had to do more work to really say which is more susceptible versus that's not what we are seeing. At least the surveys we did, this is what we found, where we are seeing more numbers in one crop, one variety of hibiscus syriacus. We tend to see more numbers in the upper zone of the plant. We sample from the upper, lower, and middle part of the plant canopy, and we found in the upper canopy, that's where we found more bugs. We were curious whether they moved to the neighboring plants. So in this case, where barley hibiscus was there was whitetex on one side and Ibelia on the other side. And we sampled leaves from both, and we didn't find any leafhopper colonizing these two hosts. Similarly, we had chiffon, that is one hibiscus, another block, and roses was besides that, and we sampled from roses as well. We didn't see anything there. That's the good news. That means that if there's hibiscus there, they tend to stay in the hibiscus itself. They don't tend to like to move to other hosts, neighboring hosts, especially roses, whitetex, and Ibelia. That's as far as we know. We also looked at looking at yellow sticky cards, where we stick the yellow sticky cards along the edge of this block, and you can see high numbers were caught in the hibiscus. These are all adults. Okay, we caught only adults in the yellow sticky cards, but you can see there are some we collected from whitetex that was beside the barley hibiscus plant, and that means that their adults are flying to the neighboring plants, but looks like they're not colonizing because we didn't find any indication that they are colonizing whitetex. Similarly, we saw activity of adults in the roses, that is adults when we put the yellow sticky cards, but we didn't see any indication of colonizing roses at this point. But remember that in the Asia, it is reported, roses are reported as a host for this pest, but we didn't see that kind of thing here to whatever this year, okay? And one other point I want to mention is that as soon as the growers saw this pest problem, they start spraying and they used insecticide applications. I'll talk about that in a little bit. And you see the population kind of went down. And in this stage, beyond October 28th, we saw the population is very, very low. That means insecticide spray really worked in this case. Okay. And damage, if you look at here, is very coincide with the number of bugs we saw. On barley, we saw more numbers compared to the chiffon, and more damage also found in the barley versus in chiffon. So it's kind of consistent that more numbers, more yellowing damage that we saw in that cultivar. Okay, so what we do, the best practice is try to get the plants from a best licensed nurseries as much as possible. When you bring into your nursery, make sure you put it in an area that is away from other plants so that you won't mix in, and watch from these plants. But you remember that they lay eggs in the midrib area, those you may not see anything when you bring in the plants, because it takes about seven days, I would say 3 to 4, up to 7 days will be good enough for the eggs to hatch, and you'll start seeing a population build up. So you look for and watch for that before you move your plants into the main area where you are mingling with other hosts. And also in applying it with an insecticide as soon as you get into a new facility will be very advisable because of this nature. Two things you can do for scouting. One is yellow sticky cards. I already showed you. We used these sticky cards in our trials. They are coming. You can collect a lot of adults. So it works, but more research needs to be done to understand what color of the sticky card and in relationship between this and the actual infestation. That we have to work on the next year or following years that we'll be doing. As far as now, yellow sticky card works, will be very good for monitoring. You can use one card by about 2,000 square feet. Okay. Another method that you can use is a sheet. Take a big sheet of, we call it bead sheet, a kind of a sheet or a kind of clothing that has a structure that you can hold on, and you can hit it with the on the surface. So if there is any bugs there, they will dislodge and fall into this the cloth sheet, and that you can, using your eyes, you can your magnifying glass, you can quantify to see whether they are present. So that is another method that you can use. So sticky cards and beat sheet method, that could be two strategies to look for this pest. So this is going to be my second last slide. Management. My colleagues, Dr. Philip Roberts and a lot of extension agents in our state, especially in the southern region, they did aggressively. They did a lot of trials. Okay, and I'm so fortunate that they did, and they came up with this list of insecticides that works. Okay, I will be paying some caution because bifenthrin, that is Talstrat P, and imidacloprid, that is dianetorpheron, and thiamethoxam, these insecticides has documented resistance in the native range. That is, we are talking about Indian subcontinent, where they're naturally present. They use these insecticides there to control. If the insect is coming from that region, they might be already resistant. So we saw some difference in efficacy with pyrethroids, especially bifenthrin, and these two products like dianetorpheron or thiamethoxam. Keep an eye on that, because in some population it worked, some population it did not provide a satisfactory control. But there are other products like Aurea, flunicamid, worked really well. We saw that it works. Expire, it is combination of sulfoxyphor and spinetoram. It worked very well. Altus worked very well, and also Talus, that is bupropion, that is insect growth regulators. So you can see there is a lot of insecticide groups, and I would say at least 3 classes or 4 classes right there that you can rotate and work on it. Okay, so it provides very good control for this. So again, this is sheer number game here. Just like Whitefly, you spray after 14 days, it's not going to work, because as residues wears down, another population moving in. So if you're really thinking about managing for a period of time, rotating and consistent application of insecticide is required in this case, especially in the fall when the temperatures are very high and the population is overwhelmingly high. Okay, so the last slide here is basically my contact. This is basically the same, the website I mentioned, and this is my phone number, and the ways to get me, through LinkedIn and X. Okay, with that, I will stop here. I know I went over the time. I'm really sorry about it, but is there any pressing questions?
No, Shimat, not that I have seen.
Wonderful. Okay, thank you so much. I really appreciate, basically, thank you so much for your patience to stay a little longer. And this basically concludes this webinar. Okay, thank you so much, Richie.
All right. Thanks everyone for joining us. Remember to send us those sign-in sheets. Get them to us, scan them, or take a picture with your phone. Send it to us at gtbop@uga.edu. If you need to mail it to us, the mailing address is at the top of the sign-in sheet. And like usual, you can reach out to all of our team here if you have any questions about the process or about the sign-in sheet. And with that, we will see everyone in 2026.
Thank you so much. Bye-bye, Bikash. Bye, Richie.